Before we start, and several times throughout the lab today, be sure to label your PCR tubes. You're going to have to label them with enough detail so that we can come back to them next week, as well as figure out which tubes belong to which groups after centrifuging multiple tubes all at the same time. So when labeling tubes, be sure to include your lab information. So in this example, it's Tuesday AM lab, so you can label it TAM if you wish. In this PCR tube, the no treatment group control sample has been placed, so there's no on it. And then you also need to label your table number. So this is table 2. So all in all, this is Tuesday AM, table 2, and no treatment group. So here in step 2, our TA is demonstrating how to move our zebrafish larvae that have been euthanized to our RNase-free PCR tube. So be sure to remember to use a wide-mouthed pipette for this so we don't squish zebrafish as we move them and unintentionally lose our RNA prematurely. This centrifuge is our room temperature centrifuge. We will only use this to spin down our zebrafish larvae prior to homogenization. This may be on one of the benches in the lab, but look for the centrifuge that is not in the deli fridge for the initial spin down. So after lightly centrifuging our zebrafish to help get them to the bottom of our PCR tube, now we are on step 4. We are in the hood and we are adding 500 microliters of tri-reagent, our pink solution, to our tubes. Following this, we're going to use our homogenizer stick to break up and free the RNA from within our zebrafish. So you'll have to repeat this 75 to 100 strokes. The goal is to ultimately homogenize our tissue completely, so that means an evenly distributed solution. You shouldn't see a large number of chunks floating around. So after completely homogenizing our zebrafish samples, we're going to place them in the centrifuge for step 5, where they will stay for roughly 10 minutes. So while removing our PCR tubes from our centrifuge, this is the first example of when proper labeling is extremely critical. If we have multiple groups worth of PCR tubes in the centrifuge, it will make things a lot easier and more clear if the tubes are labeled with specific group information as well as treatment information from our zebrafish. So now we are back in the hood for our step 6. Our TA here is going to help us remove the supernatant from our first spin down in the centrifuge. So after our first spin, we will see this pinkish supernatant, and currently this is where our RNA is. So we want to very carefully remove the liquid on top of our pellet without disturbing our pellet and transfer it to our new set of labeled PCR RNase-free tubes. Immediately following the transfer of our supernatant, we're going to add our chloroform and then vortex our samples. Here in our samples, you can see a clear layer of chloroform on top of our tri-reagent, which is going to be thoroughly mixed after vortexing our samples. So this is all in step 7 and 8. After our next trip to the centrifuge, if you look closely in our sample now, you will see that it is separated out into 3 separate layers. So on top is a very clear layer, and then a murky whitish layer, and then on the very bottom, our pink trizol or tri-reagent. So you need to be very careful not to disturb the separation of materials. And in this next step, we are trying to remove the clear layer from on top, which is where our RNA has separated into. So if you see Dr. Small's example here, you can very, very carefully pipette the clear liquid off the top, and then you're going to transfer this into another clearly detailed and labeled PCR tube. You can repeat this process a couple times, as you've seen Dr. Small do here. Essentially, you can repeat pulling the clear layer on the top off once or twice or possibly 3 times, all the way up until you inadvertently eventually pull up some of the pink tri-reagent as well as the white cloudy layer. So one trick to resolve this is allow the liquids to separate out in your pipette tip and then simply dispense the cloudy liquid and your pink tri-reagent back into your pink reagent RNA tube. And at this point, you are done pulling the supernatant off this sample. And here is a slightly more close-up version of Dr. Small's extraction. Thank you, TA, for helping us. And again, this will complete step 9 once we pull the rest of the clear layer off of our sample. So hopefully by now everyone has had some practice in resuspending pellets and spinning down pellets. We are going to fast forward down to step 13. So the tubes in this image have been spun down with our 80% ethanol. We have removed as much ethanol as possible without disturbing our pellet, and now we've placed them upside down to completely dry. And this is the end of the video for today.